We have the pleasure of having Dr. Manish Sagar, who is currently at Stanford, the University School of Medicine. He directs the Brain Dynamics Lab in the Department of Psychiatry and Behavioral Sciences. And he is a computational neuroscientist who's trained in machine learning, neuroscience, and psychiatry. And he is very much a fan and contributing to novel approaches to try to unpack the dynamics of connectivity, of brain activity, and move into a space which we already know we should be in, which is that we know the brain is dynamic. We should be looking at it with methods that can capture that at the whole brain level. And so today he's going to be talking about precision dynamical mapping using topological data analysis, and he's going to be looking at hub-like transition states in resting data. And so without further ado, I will pass it over to you, Manish, and thank you very much for joining us today.
Thank you so much, Vince and Armin, for having me. It's a great pleasure. And like you said, we badly need this community. So thanks so much for spearheading that brain dynamics community or brain space community. Okay, so today I'll be talking about this recent paper that we just released on bioRxiv, where we used Midnight Scan Club data to look at how the brain at rest organizes or doesn't, and to look at the rules and the properties that govern that kind of organization. Okay, so without further ado, let's start. So here is a mission statement slash motivation for this project and other projects that we are doing in the lab. Just to give you an overview of why we are doing what we're doing. And so the idea is to distill high dimensional data, and that could be any. Right now we're focusing mostly on brain dynamics, but it could be, you can think of molecular data, gene expression data, any kind of data that is very high dimensional, can be somehow represented, distill it into a vibrant, basically means interactive or clinically relevant representation that is easy to comprehend, and that can be interactively explored so that we can discover new aspects of data. And the condition that we kind of impose on ourselves is anytime we do any kind of averaging or collapsing the data, we should try to avoid it as much as possible. So how can we look at the data in as much, while keeping as much the data true to its original acquisition state as much as possible without collapsing across space, time, or people at the outset. So this is just another cartoon way of saying what I just said, that we are really into trying to discover, trying to capture rather the shape of the data. And I'll explain in a second what do I mean by the shape. But if we used to be doing, these days we're not doing as much, averaging across space, time, or people, but initial studies of neuroimaging were kind of sort of living in this cube space. And then we plan to move to this space where we're looking at within session data as close to the acquisition rate as possible, and without really having any limits on should we parcel the data or not. So kind of sort of, maybe it's a cliche at this time, but kind of sort of going as much data driven as possible. Now to do something like that, we have to have some way of capturing that shape, the so-called shape that I mentioned. And so these days we have been very much interested in this area of topological data analysis. And a big thanks to Gunnar Carlson, who was my mentor here at Stanford, who is still my mentor. He came up with this whole, I mean, he's considered one of the fathers of TDA in a way, because he's the person who started and kind of sort of brought it from the theoretical perspective to practical, so we can actually apply some of these mathematical concepts. And the idea is very simple. So for example, if you have some kind of two-dimensional data, like you sample a circle with some noise in it, then if you show this to a human, they may right away say that, yeah, it looks like you're sampling from some kind of loop. So how do you capture that insight? What kind of algorithms do we apply, not just in two dimensions, but high dimensions, that can kind of sort of give us an idea at a very broad level? Yeah, it looks like a loop of some kind. And that is the kind of insight that TDA type analysis are looking for. And there are multiple topological data analysis methods that people can apply, from persistent homology to mapper. And today we'll focus mostly on mapper, but we're also working on persistent homology. Maybe next time I'll tell you more about that work. So mapper is one kind of analysis under the umbrella of TDA. And just give you another toy example of how that kind of works is, imagine you have a three-dimensional data, in this case, it's a favorite mathematical object, a trefoil knot, which is essentially a loop, but it's represented as a three-dimensional, high-dimension space as a knot is where an ant starts to walk or some kind of insect starts to walk on it, it can keep walking without falling off. And so if you don't know about the shape of the data and you're trying to understand whatever this high dimensional data is, how does it look like in some kind of lower dimensions that we can interact? I mean, in this case, it's only 3D, but if it's just to get the point across, I took a simpler shape where we already know the shape. But if you do some kind of dimensionality reduction, let's say from 3 dimensions, you just go down to 2 dimensions, we get this kind of point cloud. And I'm keeping the coloring just to, again, I'll tell you in a second why. And this is where most of the, let's say, dimensionality reduction approaches stop, which I'm calling filtering, but you can call it dimensionality reduction, like TSNE, PCA, any kind of thing. Where if you go to lower dimensions, you get this point cloud, and you can do some kind of kernel density estimation and all that things out on top of it. But there is not any particular momentum to capture the shape as some kind of representation, overall representation. And that's kind of sort of done in Mapper by following additional steps. So first step is just to do some kind of binning. And you can do ND binning, like some dimensional binning, or you can do binning in some kind of k-nearest graph as well. And once you do the binning, then this is where kind of the magic happens, or this is where this step called partial clustering is where Mapper tries to retrieve the lost information or some aspect of lost information back into the low dimensional space. So for example, here, if you look at this particular bin where the crossover happened between green, blue, and red, what Mapper tries to do is in each bin, it tries to go to the original high dimensional space, in this case, just a 3-dimensional space, and then cluster the points in the low dimensional space based on that information in the high dimensional space. So if in higher dimensional, green is, we know, above blue and red, like it's farther away, and blue and red are closer, then it'll try to preserve that information in this partial clustering step. And then the last step is graph construction, where because we have overlapping bins and we want to construct a shape, some kind of, in this case, a shape is a graphical representation, we try to connect the dots, connect the bins if they share data points. So here, because they're overlapping bins, more likely than not, they will be sharing points, and then we create a graph out of it. But because of this partial clustering step, just reiterating here, we get to bring in some information from the high dimensional space that we have lost because of dimensionality reduction. And what then ends up happening is this: we just take this loop, it's a graph, we just expand it, just basically literally drag it, and some kind of force layout, you see that it captured the shape, which is a loop, which in other algorithms which leave you at the point cloud, you just don't get that idea of shape. And once you get the graph, you can do all kinds of insights on top of it that you otherwise lose if you stay in the point cloud space. So Mapper gives us a couple of advantages. One is we get a graph and we can study it, but more importantly, it has this partial clustering step that tries to preserve or tries to bring in at least some aspect of high dimensional information into the lower dimensions. And we applied this earlier on fMRI data with task, just to, this is an older paper where it essentially took all 4-dimensional data per person, did the whole filtering step. We used a nonlinear filtering method similar to TSNE to do the first step, and then when we did the binning and collapsing, we used partial clustering in the original space again to capture as much data, as much information from higher dimensions here, and then we connected these blobs to create a graph. And then even though it may look like that we are compressing data in some way, we don't really average the data. It's just that these TRs, you can go back to them. So it's lossless compression in some way. So you can go back to this node and say, okay, which TRs belong to this node, and you can color the graph based on these things and so on and so forth. So that's the basic idea of Mapper and how we applied it to neuroimaging. And let me very briefly take you through the evoked. So here are some of the applications. And I'll today focus only on these two, but I'm putting these 3 just for future reference that we are applying. So we started with looking at fMRI evoked transitions. This paper that I'll talk about today mostly is all about resting state transitions. But now we are applying these methods, now that we're more confident in them, applying these to looking at interpersonal interactions in hyperscanning or decoding states while you're watching naturalistic stimuli, or obviously looking into psychiatric populations and intervention of all kinds to see how the representations change and can we learn from them. So with the evoked fMRI data set, this is where we use Peter Banatini's data from his lab where Javier Gonzalez Castillo did this experiment where they had people come into the scanner, lie down for about 25 minutes, and while they're lying down, they were given multiple tasks in block design, but there was a continuous multitask paradigm, meaning they went from one task to the next within one session without stopping the scanner. And the good thing about this is, it's a great idea because they wanted to somehow mimic ongoing cognition in a controlled way. And it's also good because we know, the whole paradigm is awesome because we know exactly where the ground truth transition is supposed to happen, meaning from one task to the next, what kind of transition at what point happens. We kind of had to have a ground truth as compared to resting state. And also, the other cool thing is we know precisely how the person performed in every single block of this these tasks. So then we can kind of relate it to the properties we see to some aspect of behavior. So here's one person's data, again, 4-dimensional, we have about 1,000 TRs, and we kept it in the original voxel space for this experiment. And you get this kind of spider chart when you run mapper on it. And these spider charts, again, each node is representing whole brain maps, which are somewhat similar for some definition of similarity, and then end up becoming represented by one node. But the other cool thing is, and the size of the node tells you how many TRs, how many frames end up collapsing under that node. And we can color these graphs because we know precisely at what point, what task you're supposed to be doing. And this is just one way of coloring. You can annotate the graph in different ways. And when we color, this is just one person data, but when we color it across people, we find that resting state, this green color usually ends up in these peripheries of the graph, whereas when you're doing a task which requires sustained attention or which requires that your brain patterns are stable across the task, for example, NBAC working memory task or math task where you're required to focus or do the task properly, those TRs tend to be collapsed in the core. And that kind of makes sense if you think about it, that when you're doing a task, when you're very focused, your brain state or brain activation patterns are not changing as much. So they are very similar to each other, much more similar than, let's say, when you're doing mind wandering during resting state, could you be thinking of all kinds of things. So this core periphery structure was observed as a generic topological property of the data. And by the way, we see it in some video task as well, because the video task here was looking at an aquarium, and whenever you see Nemo, that fish, you press the button. So it was kind of sort of similar to some degree of resting state behavior. And we represented each node as a pie chart because, again, we wanted to keep as much information as possible about where each TR is coming from. So a pie chart simply tells you the TR that become part of this node, do they belong to what kind of task? So just give you a proportion of TRs coming from different tasks. And because we did it at single subject level, these manifolds, these graphs, like every state, every configuration is defined for a single person. There is no group state defined here. Then we could look at these individual manifolds and see whether they are behaviorally useful. And in this case, I show you two charts. I don't think I can do polling here, but if you may have already seen the paper, but one of them is the best performer on the task, and the other one is the least performer in terms of behavioral performance, which is not like very bad, like 70% versus 95, 94% or something. And you can kind of sort of get some idea there, because here you see the tasks are, so the shape graph representing the task is much more modular, where the red nodes prefer red nodes, connectivity, yellow is yellow, and orange is separate. Whereas, so this basically tells you that for this person, the spatial configurations or states that they were using were very task specific, potentially showing you high task separability, end up becoming one of the best performers. Whereas this guy who is kind of sort of all the task states are merged together in some way, like there's not using task specific activations or networks or representations, end up looking like a big pie chart of everything together, is kind of turned out to be the worst performer. And the idea here is that then we basically took this simple measure of modularity on these graphs, where it's calculated based on this task annotation to see whether it relates to behavior, and it does relate to behavior very nicely in terms of percentage correct response time and even just percentage missed. And further, we kind of made this tool, which you can, if you're interested in this kind of analysis, you can go to this link. We're calling it Dinosaur, Dynamical Neural Spatial Temporal Representations. But you can essentially play with your own data, and then kind of it creates something like this. This is one of the older versions written in MATLAB. There's a much better version in Python now. But once you get this shape graph, you can also go through some kind of movie. So, for example, here, sorry, I'm going to move my slider out. But here below and the bottom of the screen, this is a TR. It has increasing TR and basically shows you, it just highlights where that TR is. And because if you remember this mapper, there's a lot of overlap due to binning. So you end up seeing the same TR as part of multiple nodes because of the overlap, and because they're connected, they must be sharing TRs. And that is kind of sort of what gives us this comet-like tail moving on the graph. And the second thing is, which is actually very important, is because of that overlapping, for each TR, we could look at the other TRs which are highly similar to this TR, and then create these spatial mixture models of activation deactivation to look at significant activation deactivation at the single TR level, which is usually very hard to do. But because of that overlap, we can do it. And as the movie progresses, you will see the first state that the person is in resting state. It'll start somewhere here. So as this person is doing resting, it's flowing on this manifold. You can also see the default mode network coming and going. And then they will check in with the core, still resting state, did some more loops. And then they will cross over the blue state, which is instructions. The next start is starting up. And now the person is going to end up spending much more time within this core, because this is the working memory task representation is supposed to be much more similar. You can also like annotate the same graph by hit or miss, like where they made a mistake versus not made a mistake, and do all kinds of things on top of it. And the last piece from this last paper is that we could then also look at transitions. So we could capture these recurrence plots out of these graphs. So once we went from high dimensional data to creating these graphs, and then from these graphs we can go back to creating a recurrence matrix, looking at every TR to TR, how similar each TR is to every other TR. And from that matrix, you can easily just collapse it, just do a row sum or column sum of that matrix to then get transitions over time. And this is average across all subjects. And without knowing anything about the task structure, we could see that these transitions very nicely flows as per the task design. So it kind of captures the dynamics very nicely at the single TR level. Okay, so that was all task. Let me tell you some new stuff, which is looking at the intrinsic activity. And as all of us probably know, the rest is far from restful. We get to think about way too many things when our mind is wandering. And so we wanted to see if we can, and there has been so much work looking at the resting state, but we thought that although we understand to a large degree like what kind of dynamics are happening in the resting state, for example, we know the brain is somewhat bistable, and then we can look at some kind of states using different techniques. But we wanted to see if we take the data in this whole high dimensional space without collapsing in space-time, like no sliding windows, no looking at the connectivity, look at the activity itself. So rather than looking at functional activity, if we stay at the activity level, can we capture the underlying structure, some kind of manifold that is supposedly governing these transitions? And maybe there is none, maybe there is no one, maybe it's all random, but even then we wanted to see what kind of structure is there, if there is any. And is that structure stable across sessions, meaning the same person, can you identify the person based on their manifold or its topological properties? And then we also wanted to figure out if you go from, let's say you define some definition of state, in our case, if you're on one side of the manifold in the intrinsic landscape, and you go from one side to the next side, or one kind of state, so-called state, to the next state, can you jump directly, or do you have to pass through some other state as an intermediary or washout period state or some kind of state? Is the direct jumping possible, or you have to go through these intermediate states as well? So for this analysis, we looked at already collected awesome data sets rather than collecting it ourselves, and we found this Midnight Scan Club data, thanks to Nico and Damien Fair, who were also part of this paper, that we had access to this pre-processed in their best way that these guys pre-process data. So it's already in its most pristine form, I guess, to some degree that we can get. So we had roughly 5 hours of resting state data from each participant, collected over 10 sessions. Another cool thing about this data set is the parcellation is individualized, meaning for each person, these parcels were defined for that person, and it's shown in their previous paper. These parcellations change across subjects, so it's not always, you can't really just use a group average parcellation because there's sometimes large differences in these parcels. So in this particular paper, rather than sticking to the vertex level or voxel level, the features or the input to mapper was individualized parcels. So we used it because they had this nice individual parcellation. So we kind of stayed to this slightly lower dimension rather than going to 300,000 voxels or whatever, or surface vertices. We went to about 600 parcels. And then we applied mapper approach again, similar to the evoked task fMRI data. And we did split data validation because we had 5 hours of data. We kind of thought it's too much, so we split it into 2 pieces. So you will see odd and even sessions. So out of 10 sessions, we have 135 the odd sessions, or 246 the even sessions, and we get 2.5 hours of resting state data each that we tried to use as discovery and replication set. So whatever we find in one, do we see the same thing in the other? We also use null models just to see whether the properties that we observe, topological or topographical properties of these landscapes, are they simply driven by linear properties, which is fine if they are, but like, can they be linear properties such as autocorrelation or covariance structure, or is there some nonlinear aspect of the data that can be captured using TDA-based approach? So we use these null models as a comparison to see the properties we see are different from multivariate AR or phase randomized null models. And then we also replicated this in the human connectome project and equal to 100 unrelated cohort. So this is just a one subject's one session data, but we, as I mentioned earlier, we did data split. So for each person, we had 5 sessions, 2.5 hours worth of data in their individual parcel space that we ran mapper on. And again, each node in the mapper is telling you, is basically representing different whole brain topographies, but that are very, very similar. And the size of the node varies a lot because the data we have is much higher length. So, but it can go from like 5 TRs to 250 TRs. So one of the things you can do when you get this kind of manifold as a network is you can right away start to look at the nodal properties or the degree distribution or some kind of topological property of the data. Because we don't have, like in task state, we had exact tasks that people were doing, so we could look at we can annotate the graph based on which task they are doing. Here, we don't have that kind of information. We don't know what they're doing, so we can color it different ways. And one way you can color these graphs is just by looking at the nodal degree. What's the degree of each node? And it turns out there are some high degree nodes and some not so. And you look at the degree distribution as well, and found this kind of bump in the degree distribution that was kind of interesting to see. And then we compared this bump with null models. So average across these 10 people for odd or even sessions separately, we found that if you look at the degree distribution, it has a fat tail. So the real versus null data has this high degree nodes in the real data, which are not there in the null data. And that kind of sort of piqued our curiosity as why is it happening? Why should we have this fat tail in the data? And is it some nonlinear property that is being captured here? Fat tail distributions usually imply there is some kind of hubness in your manifold, in your graph. So then we combined this high degree nodes with the concept of high centrality. So we said, okay, show me the nodes on the graph which are high degree as well as highly central. And then you get this kind of hub nodes on the graph, which is just shown for one person, odd versus even sessions. So even though the manifold looks kind of not exactly the same, but you see these hub nodes at the center, which were popping up, and they were there in pretty much all the participants. We could see these hub nodes. So one interesting property is that as compared to the null, the manifold has these hub nodes. And what are these hub nodes and what do they actually mean? So to further understand these hubs, and to further understand the whole manifold, we had one more trick up our sleeves, which is instead of coloring the graphs by the topological properties, can we also color it by topographical properties? Meaning, can we color by each node by looking at the TRs that it has and just see if you look at mean activation for each of these networks that are now individually defined for each person, can you, if you z-score that average network activation, then can we see which TRs have above, 0.5 standard deviation or 1 standard deviation above mean activity in each of these TRs. So if you take one node and it has a bunch of TRs in it, let's say 100 TRs, then how many of these TRs have above 0.5 or 1 standard deviation above activation in the default mode or some other network? And that's how we used to color this network. It's just another way of coloring it, annotating it. We didn't use this information to make the graph. It's just to color it to see where things are. And when you do that, it's for the same person again, you kind of sort of, maybe I'll zoom in the next slide, where you can see that there is, it's not like one network pops up and the others just are silent. It's a lot of this, all networks go together approach, but there are, if you look closely, maybe if you go to the next slide, if you look at this one person manifold from like odd sessions only, you will see that the red, which is the default mode network, has kind of sort of, actually this for some reason looks like map of US, but the southern border or the, maybe has its default mode network more in majority, whereas if you go up northeast, you have this ventral attention network, which is this blue color, is more dominating in addition to some visual network. And then if you go on the west coast of this manifold, you have a lot of auditory singular percular network as dominating. So what we briefly see, and then if you go to the hub nodes, they look very interesting in themselves. The more central nodes are kind of sort of uniformly representing all networks. It doesn't have any particular network dominating or one or two networks dominating. And we then looked at each of these nodes, and if you just look at one node, again, you can look at the spatial profile by either doing spatial mixture modeling or just averaging the TRs within that node. And we found that, lo and behold, it has this properties of the default mode network, more or less, but not just default mode, but other networks are also kind of sort of part in it. And then you can also look at the, for each network, the z-scored value. And so this is the band of like 0.5 standard deviation, and DMN is kind of leading highest in this particular node. The TRs have higher activation in the default mode regions. And if you look at some other area like the top left, top right, then you have ventral attention network leading deactivation, the TRs where ventral attention was high. If you go to the left, we have the auditory network leading, and others are kind of following and staying within this band. But when you look at the hub nodes, that's where we found this uniformity across all networks, that there is no one particular network leading on average. All the TRs are kind of sort of somewhat equally contributing or staying to the mean. So zero basically means mean activation because they are z-scored. So one finding we found was there are hub nodes. Second thing we found that on this manifold, if you color them by resting state networks, you can see this topography of some networks dominating on some part of the manifold, whereas other networks highlight or dominate on other aspects of the manifold, other place on the manifold. And you can then try to quantify it. So you can see, are these different networks being dominated, are they subject specific, or is it like more subject generic effect that everybody has default mode in the south or something like that? And to do that, we did very simple. I mean, if you want to do it at a single node level, it may cause what we call a graph correspondence issues, because each node you have to then of a manifold compared to every other node and every other subject, and you have to solve the graph correspondence problem. But we kind of simplified it by looking at for each network, wherever they're highlighted, for whichever node has whichever network, are there other networks that go up or down? So we created this co-localization matrix, which is just showing which networks go up, go together, activate together, and on the network. So DMN and frontoparietal, for example, this person kind of go up and down together. So you have a high similarity between DMN and frontoparietal network. And then we organize this whole similarity matrix by looking at higher order versus sensory, just to ease, just to clarify what may be happening. And when you look at these co-localization similarity networks, meaning which networks are up or down together on across sessions for each subject separately and within a subject, and we found that if you look at within subject, the similarity matrices are very, very similar. So for example, in this case, these are odd and even sessions. You'll see default mode network here, ventral attention on top, auditory on the right-hand side, or left-hand side for even sessions versus odd sessions. And the matrix captures that pretty nicely, that these two matrices are highly correlated, as opposed to this another subject number 2, who actually has a dominance of the yellow color, which I think is the frontoparietal network, versus default mode versus some other somatosensory network. And they also show up again when you look at their data again with different sessions. So when you then compare the similarity between these co-localization matrices, it turned out they're highly similar within the person, whereas between persons, like correlation values up to 0.9 and above within the person. There's very high subject specificity, even for subjects who have apparently slept in the scanner. So I remember in the Midnight Scan Club data, you may remember that subject 8 slept in the scanner, didn't have good data quality, but even there, this property held up very nicely, that they had very high within subject similarity. I'm also showing like the group average, which kind of sort of disappears at that point. And the other thing is, if you look at, in the anatomical space, there is this whole gradient research, like the topological gradient, something like that, where the idea is that the higher order networks kind of sort of hang in together, whereas the unimodal networks are separate, and you get this differentiation between the two, which is very interesting in itself. And we kind of see it as well only if we look at the central tendency, meaning if I average over all people's co-localization networks, I get the similar plot that higher order are more tightly knit than the unimodal ones. But if you look at the individual people co-localization matrices, that's not necessarily true. So with this kind of analysis, we can get much more fine-grained information about that person rather than getting the whole gestalt average, which may be true on average at the group level, may not be true at the single person level. Just a side note. Okay, and then the dynamics piece finally. We want to see how does if you traverse the map of the graph itself, the manifold itself, what kind of properties can we discover? And then secondly, what if we then take it back to the time, the original TR space, time domain, and does the traversal has any interesting properties? So I kind of mentioned this earlier already, that if you look at the nodes which are dominated by one or two particular networks, so here I'm going to show you 3 different trajectories. The first one is where default mode network is highly activated on the side. But as you go closer to the network closer on the manifold towards the center of the manifold, you will see that domination of default mode network goes down to the point where when it reaches hub nodes, there is not much any one particular network leading the way. So as we traverse on the graph from the periphery towards the core or towards the center of the graph, we see that that dominance goes away. And one way to quantify that is you can look at entropy or some other cool measures, but we did it very simply by saying, okay, if you look at the mean value of each of these networks, is there a variation in the mean? If you look at the peripheral node, the variation is pretty high because the means vary a lot across networks. But as you go toward the center, the variation disappears, and they're all kind of sort of sticking at z-score value of 0. So the mean variation across means goes away. And we just use that property to then color the graph next. But the same principle holds for other networks. If you look at auditory network, if you start at the periphery, auditory leads, but as you go closer to the hub or the center of the graph, they kind of sort of go towards more normalizing and everybody staying at the mean value. But when we take this variance as a coloring scheme where we say, okay, the high variance is for nodes which are on the periphery, where as you go towards the center, the variance goes closer to 0 or the standard deviation, then you get this kind of nice looking topographical gradient. So which is truly a gradient in this, because you have, from calculus perspective, you have the variation going down to a point where there is no variation in the hub space. So it's for the traversal on the manifold, we get these nice gradient looking shape of the manifold, and this was kind of subject generic across all 10 subjects, and then later on the human connectome data across all subjects, we found a very similar gradient where it did not differ across subjects, meaning this variation across the manifold was similar, and you get very similar looking gradients. And now let's look at traversal in time domain. So how can we now go from or look at the traversal instead of looking at the traversal on the manifold, which may or may not be following the same way in time? We can go from each manifold back to individual TRs. So essentially, for each node, you have a bunch of TRs, And in this case, just for the simplicity's sake, I took the majority node. So if, in this case, if the node has default mode network as the majority, then the TRs within this will get the default mode label. If you're looking, if I'm kind of unpacking it at the TR level. And I had to define a new state because hub states don't have any majority. They pretty much all of the networks are similar. So I just defined a hub state for the nodes, which are hub nodes, where the activation is similar across all networks. So I just call them hub. So you have same, again, 10 or 12 networks. Node just means none of the above. So whatever reason, if we use a threshold like 0.5 standard deviation or 1 standard deviation above the mean, and if no network wins there, we are calling it nota. None of the networks show up. And then so hub state versus all the other networks. And if you look at the individual TRs, this is kind of a bar chart now because each column here is one TR, and you can kind of get an idea that these gray looking hub things appear a lot. And we can look at all 10 subjects or in even sessions, only showing like the first 600 TRs because we have about 5,000 or something like that TRs, maybe 2,500 TRs per person, and gets very messy. But if you look here, you can get an idea that for same subject, similar looking states are popping up, and then this hub thing is popping up again and again. And we can quantify this by simply doing a very basic, simple Markov chain quantification where we simply count the frequency of each state and the transition probability for each state. And we get this asymmetric matrix where no matter what state you start from, and I put the diagonal to 0 just so we can clearly see the transition. And so for odd or even sessions, we see that the hub states tend to be the most sought after destinations. So no matter which state dominated network you're in, you tend to go next to the hub state. I mean, it's not very high probability, like 0.16 out of 1, but still pretty high probability to have hub states to be the dominating states. And that pattern repeats across sessions. And then you can also represent it as a Markov chain plot where you find the highest in degree. So now the nodes are colored by the in degree, and hub is the highest in degree, for example. So it kind of sort of, and also replicates. So hub states were observed to be central in time as well, meaning that these are the states that are intermediary being passed through every time the brain state transitions from one dominating state to the next. And interestingly, even these Markov chain plots are very subject specific. So we found high similarity within participants than between participants. So not just that the manifold co-localization was very highly subject specific, but also these Markov chains that we get are highly subject specific. Okay, so I'll give you a summary that our results suggest there is indeed a stable structure. There's some kind of interesting looking manifold that we find. And this manifold had this large attractor-like basin, which we're calling hub state. And that looks like that it performs this function of intermediary between different states. We found a gradual evolution of brain configuration as opposed to discrete switches. So it was like this topographic gradient or temporal gradient that we find. And then there is a huge consistency across subjects in terms of how the states are defined. So some people may have DMN as their dominant, or DMN plus some other network, and some other person it could be visual plus some other state, some other network as their dominant state. So this method allows us to look at states, state definition, state configurations independently for each subject rather than doing some kind of group analysis to find those states. And also the traversal in time is very highly subject specific, and the hope is that down the line, hopefully, when we're looking at ABCD data now, that we will be able to find some clinically relevant markers that follow these dynamics that we pick up. And for future work is, as I mentioned earlier, we're looking at hyperscanning data or decoding states or changes in landscape associated with different psychiatric disorders or pharmacological interventions to see if this mapper-based approach can capture those trait transitions as well. Okay, with that, I want to thank everyone. Thanks the funding, lab members and our collaborators, and a shameless plug that we have multiple postdoc positions available thanks to a recent grant. And thanks again, Vince and Arvin.
Thank you. Thank you. That was great. Everybody clap. It's hard to get that feedback in the virtual environment, right? We need to do better at that. Now we have a little bit of time for questions. You can either use the chat box or the Q&A option in the WebEx portal. So please go ahead and submit your questions, and we can either have you ask them yourself and unmute you, or I can convey the question and we can have a discussion about it. So while we're waiting, I was curious. This is really neat stuff, and I really like, I mean, I'm obviously very much a fan of the reduce as little as possible or do the reduction as late as possible, right? Kind of approach.
Yeah.
I was wondering, because I know you looked at the in the midnight scan data, you had you had now individual level parcels. To what degree have you thought about time varying parcels, for example? So spatially varying nodes, or even, you know, you can even think about this is some work that Armin's been doing, sort of voxel wise, you know, very complex changes. There's always, I think of it kind of like layers of an onion, right? So we have to pick our battles and dig deeper and deeper into this complex data, and it leads us into some really interesting places.
Yeah, that's a great question. Yeah.
Yeah. So anyway, I was just wondering if you'd thought at all about kind of, could you extend this work in that sort of a direction, and if so, how would you do that?
where the parcel itself changes, right? So where
Right.
the
Yeah.
parcel
Yeah.
changes over time. Yeah, that's a very cool point, and we haven't done anything in this paper, but one of the things I've been wanting to do for a long time, haven't done it, or maybe I'll do it with Armin, is that you transpose the matrix, right? So rather than looking at time, we can look at spatial configuration. So the input to the map could be just where rows become parcels and time becomes features, and then we can look at spatial configuration or the anatomical configuration or the parcel definition manifold created from the mapper. That would be pretty cool to see, and then we see whether that partial configuration changes over time. So right now we're looking at how temporal things are changing, like how the time is changing things over states, but we could transpose the matrix as input, or maybe create a new mapper which has like a tensor input where you have space and time somehow going in together. That would be pretty cool.
Mm-hmm. Yeah, neat stuff. I just realized from your bio, by the way, that you had interacted with Alan Reese, who I overlapped with at Hopkins when I was there. So
I didn't
small
know that.
world.
Yeah.
Yeah.
He was my postdoc mentor. Absolutely. Yeah.
Yeah.
He's the reason I'm still here.
Yeah. Small world. So.
So we have a question.
Oh, okay.
So Ash is asking why the hub state is the state to go into, like what's it going to a state?
Yeah.
And related to that, my question, so I'm asking two questions at the same time, is if you look at those hub state, do they have a high activity or low activity?
Yeah, both great questions. So we don't exactly know why hub, like why in the world is hub even there. I can just have some guesses, and I think the guess is that actually let me expand a little bit on that. So hub versus non-hub, right? So you have this biostability, and that previous papers have also, like Michael Breakspeare's work and Luca Cocci and other people, maybe you guys also have said about this biostability feature, right? So the one cool thing about this approach is that you can look at biostability at the crude level, but the fine grained level, that biostability can be finely grained. You can look at default mode based or some other network based states. And so to me, I think the hub is kind of doing this washout period, but there is something we have to check, that if we go from one dominated state to the next, the brain has to maybe like we will need some PET data, I think, but to look at the metabolics, that maybe hub state is this washout rest period the brain has to go to the next dominating state, because we find, to answer your question, Armin, is that hub states are the ones which are low activation states. Here, the non-hub states are where some network is dominating the above certain threshold of z value or whatever, whereas these hub states are the one where the activation stays closer to the mean, whereas the peripheral states are where the activation goes above or below the mean significantly.
And another question, if you can elaborate more on how your approach captured that topological pattern that you were referring to, spatial topology.
Like if you look at the spatial configuration. So the idea would be, let me see if I can go back. Okay, are you still seeing the?
Yes, we do.
This initial figure, right? So if the idea would be if you just transpose this matrix, so instead of having parcel, the rows are TRs by parcel, we just transpose it. So you have parcels on the rows. And now you will see these nodes will be which parcels are consistent across time. So rather than saying which brain configurations are consistently popping up across time, you will see which parcels are kind of going together when you're looking at across time. And you can color the graph by, let's say, early TR versus later TRs and see where the configurations are changing, if any. That makes some sense. So just transposing the matrix right now is giving us over time how the spatial topography is constrained. But if we change it, then the first easiest thing would be like for each session, we just give the transpose matrix separately for each session and see where the spatial configuration remains the same. Meaning, is it that the parcels or the voxels or the surface-wise vertices, are they hanging together or changing across sessions? That would be the first place to start, I guess.
So another question is there a state that is ideal in accordance with the free energy principle?
Yeah, I'm not sure what that principle means exactly.
Me neither. So Ash, if you like, I can unmute you to ask your question. So another question is
the free energy principle is that's Carl Friston's Markov blanket approach, which I think would take several weeks to unpack. So.
Is it like no free lunch kind of thing?
No free energy, no free lunch. Yeah.
So yeah.
Pegged it. Yeah. No.
So the next question from Ghetto is, when we transfers for analysis to prediction, could these TDA, topological data analysis, be advanced to predict the brain dynamic?
Yeah, so that's a great question. One of my grad students, Caleb Janice, is trying just that can be used, these manifolds to decode states. So like a very easy application is where you create the manifold during resting state, and then when a new state comes in, rather than recreating the manifold, assuming you trust your manifold, you have enough data to do it, then you can just see which node is it highly similar to. And that way you can get that decoding. So let's say if you have something generated from naturalistic or TAS data, these manifolds, and then you can, for the new TR that's coming in, you can say which the spatial configuration is similar to which part of the manifold the most, and highlight it by that. And then you can kind of sort of decode or try to decode the most simplest way. There are much more complex way you can do it, but that's a great starting point to see decoding is possible. Yeah, and he's trying that.
And a relevant question. So most of the thing you show here is mostly like showing the subject specificity, right? How are we going to go to group inferences and like doing at the group comparison?
Yeah, so we kind of shied away from that, but yeah, you're right. In some cases, you have to look at heterogeneity. So one simple way of looking at it is you create these manifold single subject level, then look at the topological or topographical properties and see where are there people who have this kind of degree distribution similar to others. Or you can do much more detail where you do graph comparisons. That's where I mentioned the node correspondence issue. But if you resolve that, you can look at, are these graphs somewhat similar? So you can either basically group people at the topological property level. So people who have the fat degree nodes are on one side, people who don't have it on the other side, or you look at people who have DMN as a dominant state versus other, or people who have a high number of hub states or not. So some property of the manifold, or you can directly compare the manifold. But there you have to solve the graph correspondence problem because each node in every person could be a different node. And the third thing you can do is, we haven't tried, we have thought of doing it as layered mapper. So essentially where it becomes a nesting of mapper. So you have mapper graphs, and then the next mapper is input of the next mapper is mapper graph itself. So you get this deep layered, deep mapper, maybe like some way of layered mapping. Then you can do it across people, across population.
So there is a statement from Hancock saying that the hub state resembles the global state that extract with the phase locking connectivity with the LIDA.
Oh, pretty interesting. I don't know about LIDA, but so what does that mean there? Is it global state? So that can they?
So again, Hancock, if you like, we can unmute you to ask to have a conversation. But I think they are using principal components.
Leading eigenvector dynamic
Yeah.
analysis, I think is what it's called. Yeah.
Okay. I need to get it.
So let me unmute him just a second.
It's like a dynamical systems, I think. Type.
I see.
I mean, you mentioned criticality, right? So it kind of ties.
Yeah. Got it. Yeah, so maybe is he coming?
Unmute Fran
Yes.
then, yes.
Hi,
Fran is itching to talk.
can you hear me?
Yeah.
Yes.
Once again, I'm not a guy.
Oh, hey.
So the thing is, rather than working with activations, we look at the phase difference, and we're looking at cosine of phase difference, which is basically phase locking. And what you do is you extract the leading eigenvectors for each TR, and there's a whole bunch of things we can do on that. One of the things, obviously, is k-means clustering, but then you can look at what's happening over each individual subject. And what's striking is the sort of where you had those gray areas. We typically use gray for the global state. And the global state is where all of the I'm actually doing it in Vox10 voxel space. So without a anatomical or a ICA parcellation. And all of the voxels at that point are all have the same polarity.
Oh, wow.
And
So some kind
that's
of
the
global
state.
phase locking. Wow.
Yeah.
Okay.
And so what happens, what you see, because you can plot them over time, you can see the way the phase locking changes as it flows and then goes in, and the switching between the various different, I'll call them spatial patterns, because they're not really states, because it's not discrete switching. So it's actually very interesting.
Very cool. Yeah, I would love to check that out. So that's basically going away from activity and adding connectivity or some kind of co-fluctuation.
Yeah,
That's
it's based
really the
purely
next
on
step.
phase looking.
And maybe that'll answer the why. What are the hub states really doing? Are these maybe these are the global states where the brain just rests in some kind of global fashion and then go out? So yeah, definitely love to check that out.
Yeah, it's very interesting. It's very similar in terms of some individuals spend a lot of time in the global state, and others are jumping around state to state, and you can actually get correlations with the behavioral scores.
Very cool. Yeah, thanks for sharing.
So on a related note, Ali Bayram is saying, related to hub, coactivation patterns cap represent spatial pattern functionally distinct configuration of activity. Is your hub state may also resemble a cap or caps in any way?
I thought the caps, correct me if I'm wrong, but I thought caps, the idea was that you have some, it's actually co-fluctuation, not activity, but something where there's some kind of dominance, right? It has a high amplitude. So maybe if maybe I'm wrong, but caps might be those high amplitude states might be happening on the periphery of the manifold rather than the hub, but that's just a guess. Because we're in the activation space, I can't comment on the co-fluctuation piece like the other person mentioned, Hancock mentioned, that it could be that hubs may be representing some kind of interesting property of co-fluctuation, which is also the case with caps, I guess. But that's definitely, maybe the next step is to brush my knowledge of these amazing dynamical tools out there, rather than sticking in the cave, and then compare them with the mapper. That would be cool.
So my 2 cents is cap also is quite when looking at the coactivation activity, but I agree with you. I think those cap they identify are usually those peripheral in your approach. So basically they disregard, if I understood correctly, I wish there was someone who can explain, but I think they disregard those hub states. So because they are low activity, this is my understanding.
One other comment too, just on the activity connectivity piece, is there's some work ongoing by Ashkan Fageri here looking at modeling of jointly modeling activity and connectivity, through the context of sort of careful sort of signal processing methods. And what he's kind of revealing is that a lot of the assumptions we make, like if you filter an activity, that will translate immediately to connectivity in the same way, is not correct. It's because we just didn't, it was just an assumption. But if you map it out and you do the math, it's different, right? It transforms and changes. And so he's got some really nice frameworks that he's working on to link all of that together. So maybe that could be attached, right, to what you're doing to try to inform about kind of multiple domains, right?
Yeah, I would love to know more about that. Absolutely. So yeah, we kind of sort of stick to just connectivity, sorry, just activity, but yeah, we kind of left out the whole connectivity space. And just to expand it further, some of our collaborators are doing things like higher order interactions, where why just two regions? Why not like, this is where the persistent homology comes in, like look at loops and voids, but basically look at higher order interactions. Yeah, I'd love to go into that space, maybe with this person you mentioned, or
yeah. Armin's interested too, so.
Awesome.
Great. Well, we are hitting 12:02 now, so unless we have any burning questions, we can close the session. Armin, do you see anything else, or?
So there are a couple of questions, but
okay.
For the sake of time, I will share it on the Slack.
Shift those to Slack, so we will make sure your questions get answered. Thank you everyone for joining us and spending this time, and thanks to Manish for your very wonderful lecture and really interesting work, and looking forward to staying in touch and seeing where it goes, and hopefully working together on something. So
love to. Thanks so much. Thanks, Vin.
Thanks everyone.
Take care.
Have a great rest of your week. Okay.
You too.
Bye-bye.
Bye.